Welcome students. Today our topic of discussion is a general account on primitive angiosperms. The angiosperms or flowering plants, one of the major clades of extant seed plants, are the largest group of embryophytes, with at least 2,60,000 living species classified in 453 families. Their number is much greater than all other groups of plants combined together. They are the dominant plants on earth at this time and have occurred as dominant for the last 100 million years. They show great diversity in their habitat than any other group of land plants. During the past 130 million years, they have conquered practically every conceivable habitat on earth. That's from hot deserts and windswept alpine summits to fertile grasslands, freshwater marshes, dense forests, and lush mountain meadows. Although a number of flowering plants live in aquatic habitats and have adapted to the saline conditions of dry lake beds and salt marshes, relatively few species live submerged in the oceans. There has, for a number of years, been much speculation as to the origin and characteristics of primitive angiosperms. While tracing their ancestors, various groups of plants have been considered as the probable ancestors by different workers. Some believe various orders of gymnosperms like Bennettitales, Chordatails, Cycadophylicales, Cycadales, and Nettales. Others, pteridophytes like Xylophytales, Lycopodales, and Equistales. And even some others have regarded algae as their probable ancestor. Fossil records have been of little help in determining the ancestry of angiosperms. The prehistory of angiosperms remains unknown, and morphologically they are so sharply delimited from the gymnosperms that there are little chances of filling the gap in our knowledge. Courese rightly points out the missing link is indeed still missing, and Charles Darwin called the origin of angiosperms an abominable mystery. In absence of definite evidence which could help in tracing their probable ancestors. Although today a huge literature exists on the same topic, but still it is an enigma. Now, major characters of angiosperms. Extensive research has been carried out by various phylogenetists to find out the characters of primitive angiosperms, and such investigations have revolved around following points. Like, have the earliest angiosperms evolved monophyletically or polyphyletically? Second, what were the environmental conditions which favored their evolution? Third, what was the habit of primitive angiosperms? That's whether arborescent or herbaceous. Next, what was the stem structure, the type of conductive tissues, and was the cambium present or absent? What was the shape and size of their leaves? What was the structure of the flower, whether relatively simple or complex? Were the petals present or absent? And when present, whether separate or united? Were flowers mono or diclinous in organization? What were the type of pollinators? What was the type of fruit they possessed? What was the number of cotyledons in their embryos? How much of endosperm was present in their seeds? And what was the type of their seedling anatomy? However, it needs to be pointed out that the concept of primitive and advanced characters is relative and meaningless unless definitely related to a particular taxon, lineage, or phylogeny. Within a taxon, the characteristics of the common ancestry are primitive and others are more or less specialized in proportion to their departure from the ancestral condition. It's common knowledge that organs do not evolve at the same rate. Delphinium, for example, is primitive in having free follicles, specialized in its zygomorphic parent with a nectar spur. The non-rhythmic correlation of advanced and primitive characters is particularly prevalent in lower angiosperm families, for example in Raniles. But in more specialized families such as Asteraceae, Lamiaceae, the ancestral features of the angiosperms seem to have been lost. The rates at which organs evolve are obviously related to the selection pressures affecting them and to their genetic ability to vary. The different rates at which organs have evolved in the same species makes it particularly difficult to assess relationships or even to infer whether one taxonomy group is more advanced than another. For this purpose, it is necessary to assess the average advancement of as many characters as possible. A further complication is that different lineages have advanced at different rates. This means that an advanced taxon may be either more ancient or recent than another advanced group. Indeed, it may have been less advanced yet younger than another. Without fossil evidence, such cases may remain unresolved. Sporn in 1948, however, has clarified a point which has confused many botanists, including Hutchinson, in their assessment of advanced and primitive characters. According to Sporn, a primitive character is one which possessed by their ancestors and is also possessed by the present day taxa. Primitive and advanced groups may be ancient and reset accordingly. Now, traditional versus recent views regarding primitive angiosperms. Magnolids traditionally have been considered to retain the greatest number of plesiomorphic features within the angiosperms. This viewpoint has received some support from morphology-based cladistic analysis. The magnolids, therefore, may represent a basal paraphyletic complex within the angiosperms as supported by the possibly retained plesiomorphic features of laminar anthers. The non-columellate pollen of magnoliales may also be a retained plesiomorphy. But most analysts of the past several years concur in placing the monotypic Amborilla as a sister to all other extant angiosperms. Although some analysis suggest Amborilla plus water lilies may occupy this pivotal position because Amborilla has spirally arranged floral organs. Now, primitive angiosperms and nature of pollinators. Within the angiosperms, there is a general trend from primitive beetle and fly pollinated flowers towards more advanced pollination by butterflies, moths, and bees. Flowers pollinated by bees show the greatest degree of nectar, scent, coloration, diversity, and pollen production, as well as diverse specialization of flower structure. Primitive flowers tend to be trumpet or saucer shaped. For example, ranunculus, hibiscus, magnolia, malva, lavatra, cucurbita, and papaver. While more advanced flowers may be tubular, for example, symphytum, digitalis, lamium, erica, datura, and nicotiana, or zygomorphic, for example, orchis, anterinum, pisum, lobelia, viola, gentiana, wisteria, memulus, lonicera, and begonia. There are thus two directions of adaptations, from polypetally, that's many petals, to oligopetally, that's fewer petals, and from separate petals to sympetally, that's fused petals. However, simple flowers as in Commelinidae like grasses, sedges, and rushes and euphorbials may not be primitive, but derived from more complex forms in the liliales. Thus, their simplicity is secondary and a form of structural degeneration rather than a primitive feature. Now, different views on the concept of primitive angiosperms. Some phylogenists believe that the first flowering plants were upland plants that diversified in tropical or arid regions about 250 million years ago, that's in Permian period or Paleozoic era. Angiosperm fossils of that age are unknown, probably because they evolved in dry uplands that were not conducive to fossilization. Thus, the lack of fossil may be due to their limited distribution and insignificant role that they might have played in the vegetation of the world. A few phylogenetists are of the view that the primitive flowering plants were tropical mesophytic trees with pinnate leaves, and the fruits were in the form of clusters of large arillate follicles. According to Stebbins in 1974, primitive angiosperms were small, woody plants with small leaves and moderate-sized flowers. According to Takhtajan, moderate-sized, solitary, axillary or terminal flower having parent of modified bracts, leaf-like stamens, monosulcate pollen and conduplicate carpel are the characteristic feature of a primitive angiospermic flower. Another important trend is shift away from single flowers, for example, papaver, ranunculus, nulambo, towards multiple flowers in racemes, spikes, whorls and heads, for example, lamium, scrofularia, gentiana, borago, laburnum, plantago, achillea, and salix. This trend reached its height of perfection in the dipsacales, the apiaceae, and above all in the asterales. In asteraceae, the compound flowers represent a spike or whorl of flowers that has failed to expand vertically, and all the flowers are packed side by side instead on a flat head, that's capitulum. We can still trace the spiral patterns in helianthus heads, for example, where the whorl-like pattern of the raceme would have unfolded if it had elongated vertically. Now, Hutchinson believed that the primitive angiospermic flowers were medium-sized and grouped together in lateral clusters, much like those of James Wintry of the family Winteraceae. The members of this family also lack vessels in xylem. Cronquist also regarded James Wintry as primitive angiosperm because of the presence of early angiosperm features like small woody plants with simple, entire pinnately veined leaves with large terminal and axillary solitary flowers on an elongated receptacle bearing a parent of modified bracts, leaf-like stamens bearing monosulcate pollen and unsealed conduplicate carpels. England and Prandtl regarded Amentepherae, including Juglandaceae, Butalaceae, Fagaceae, etc. as the most primitive angiosperms because they have unisexual flowers arranged in catkins and their flowers do not possess petals. The inflorescences resemble the cones of gymnosperms. However, this view has been rejected because the plants of this group produce tricolpate pollen grains and possess an advanced type of wood anatomy. Taylor and Hickey developed the paleoherb hypothesis for evolution of angiosperms. According to this hypothesis, most primitive angiosperms are rhizomatous perennial herbs with simple alternate net-vein-leaved, thin textured and with anomocytic stomata and flowers arranged in racemes or sun inflorescences. Flowers have numerous to few parts. Those of the parent and androecium usually are in the whorls of tree. The filament is well differentiated from the anther and the connective usually inconspicuous. Pollen grains usually have a columnar axine. The paleoherbs include aristotales, papyrales, nymphaeales, and ceratophyllales. They consider Chloranthaceae as the most primitive family because of their small, simple flowers have an extensive fossil record dating back 125 million years. Some of the important and well-documented phylogenetic characteristics about the angiosperms that had been put forward by Hutchinson in 1959 are as: woody stems are more primitive than herbaceous stems, and herbs have been derived from trees. Trees and shrubs are more primitive than climbers and twiners in any one family or genus. Perennials are more primitive than biennials and annuals. Aquatic flowering plants are derived from terrestrial ancestors, and epiphytes, saprophytes, and parasites are more recent. The simple unbranched stem is an earlier type from which branching stems have been derived. The stem structure with collateral vascular bundles arranged in a ring is more primitive feature than that with scattered bundles in a few dicotyledons and most of the monocotyledons, and the later are to be regarded as derived from the former. The paired arrangement of leaves on the stem is held to have preceded the spiral arrangement in which the leaves are solitary at the nodes. The trilacunar node is the primitive type in the angiosperms, and that the unilacular node developed phylogenetically. Primitive organized water conductor system includes vessel-less wood, the presence of tracheates with scaliform bordered pits. They flow with long sieve elements and with groups of minute pores forming scattered sieve areas along their longitudinal walls and very oblique end walls are considered primitive than the one with horizontal end walls having a sharply defined single sieve area. The flowers of the earliest angiosperms were radially symmetrical, have had an elongated axis with an indefinite and variable number of large free parts arranged spirally on it. Separate parts are generally considered to be ancestral to fused parts. Separate petals preceded united petals, and separate carpels preceded united carpels. Such flowers are very similar to the strobili of Cycadian gymnosperms. The primitive type of stamen has broad laminar structure undifferentiated into filament and connective. The pollen grains in primitive type of angiosperms are with only one polar germinal furrow in the spore wall. Carpels in primitive type of angiosperms were large, leaf-like, partially closed at the time of pollination and containing many ovules. The style in primitive angiosperm is absent and the stigma decurrent along the suture of the carpel. The seeds in primitive angiosperm are large with abundant endosperm and a very small undifferentiated embryo. the primitive fruits appear to be cone-like structures consisting of a large many-seeded follicles. For example, an Apocarpus gynoecium of Magnoliaceae, presumed to be a primitive family, matures into a cluster of follicles. With this, we conclude today's topic, that was about a general account on primitive angiosperms, wherein we learned that angiosperms are the most recently evolved major group of plants, and they have been in existence from the early Cretaceous, that's some 132 million years ago. Second, the most significant factor that modifies the floral structure of angiosperms was their co-evolution with insects, as a major evolutionary trend with respect to pollinators have been from beetle to butterflies. And lastly, the traits like growth habit, leaf arrangement, stem structure, wood anatomy, nodal anatomy, and above all, the reproductive structures that had been put forward by Hutchinson and modified by others are to be taken into consideration while describing the phylogeny of angiosperms. Thank you.